everyone. I wanted to review a little bit of information related to today's lab exchange pathway and the lab we're actually doing in person this week. So before class, some of the initial cloning steps were done for you. So the overall idea is that we want to take a gene of interest and put it into a plasmid. Now, when we do cloning, the cloning process isn't always perfect. So ideally, we can see in the first slide that we want our plasmid to have the insert of interest. This is what we're aiming for. Sometimes during cloning, though, the plasmid will reanneal on itself and insert does not go in. Other times, something happens along the way and we don't get any product. So again, all three of these are possibilities when you're trying to clone a gene. And today you started with 3 samples that were labeled A, B, and C. One of your overall goals is to figure out which one is the good one. You're going to identify all 3. So A, B, and C correspond to all 3 of these scenarios. And in doing so, you'll figure out which one looks like this, which one is the good one. Okay. Now, if you found that good one, what would you actually do next once you had cloned your gene successfully? And a common next step would be to do a mini prep. The overall goal of a mini prep is to extract plasmid DNA. Okay? Now, during a regular non-COVID semester, we actually do mini preps in the lab. But because of COVID and the fact that we're only doing labs every other week, we actually don't have time to do mini preps this year. So one of the videos in today's lab exchange pathway will explain what a mini prep is and how to do one. So again, you'll be watching a video that will explain this process of how to do a mini prep to allow you to isolate and extract plasmid DNA. Okay. Now, after you've done a mini prep and you've isolated and extracted the plasmid DNA, what would you do with it? There are actually lots of possibilities. So one thing you could do is you could place the plasmid in specialized cells so that they could express the corresponding protein. And then that protein could be used in all kinds of lab studies, or it could be used for pharmaceutical purposes. Maybe you're expressing some kind of a protein that could be used as some kind of a medicine. Now, this entire process of cloning a gene, expressing it in certain cells, and then trying to obtain some kind of a desired outcome, that process is sometimes referred to as genetic engineering. So one of the other videos that you'll watch today on Lab Exchange is a video all about some of the goals of genetic engineering. So the video will discuss some of the possibilities and some of the potential that's associated with genetic engineering.